On a hill far away stood an old rugged cross, the emblem of suffering and shame. And I love that old cross where the dearest and best for a world of lost sinners was slain. So I'll cherish the old rugged cross till my trophies at last I lay down. I will cling to the old rugged cross and exchange it someday for a crown. Oh, that old rugged cross, so despised by the world, has a wondrous attraction for me. For the dear Lamb of God left His glory above to bear it to the Calvary. So I'll cherish the old rugged cross till my trophies at last I lay down. I will cling to the old rugged cross and exchange it someday for a crown.
St. Buckman's College, having the legacy of 101 years, was and is always in the forefront of imparting knowledge in the area of higher education. Last year, as part of our centenary celebrations, even before the official inauguration of the centenary, we started the centenary speaker series with great Nobel laureates like Anthony Leggett and other renowned scholars like Noam Chomsky, etc. And I think last year, we all remember with great pleasure that last year, from 19th of September onwards, we were celebrating the vigor of knowledge, Samvit 2.0. The seniors may remember that. And we were very much celebrating the All India Science and Cultural Exhibitions conducted in our campus. And we are proud that around 1.5 lakhs students and public visited this science and cultural exhibition. So last year by this time, this was the second day of that exhibition last year. Along with that, we had a lot of other programs also on those days. But every occasion of the celebrations were marked by some academic activities also. Thus, on the Navadi celebration time, that is on 90th year of the establishment of St. Buckman's College, we started BELLS, Buckman's Erudite Lecture Series, and by Nobel laureates. And our distinguished guest, French, Ada Yonath, was also were here in this great hall to address our students with as part of this eminent lecture series. And it was instituted by our own teachers, that means the teachers of our institution itself. And 5 of them were happened up to now. 5 great Nobel laureates made their lectures to our students and teachers. Now, in connection with centenary, one of our proud alumnus, Dr. Cheriyan Sebastian Thachenkary, who was here in our college from 1968 onwards, and he did his UG here, and then moved to US and to Canada, and along with his wife, Pauline Thachenkary, has instituted another speaker series. And for the first lecture of this Rao Sahib Dr. Joseph Thachenkary Memorial Distinguished Speaker Symposium, and it is being inaugurated today by the great scholar and world-renowned biophysicist, Professor Dr. Thachenkary Singh from All India Science of Medical Science, New Delhi. And he is inaugurating this symposium series. And doing the first lecture, he will be introduced to you soon in detail by Dr. Ranjit Thomas. As you all are aware, Dr. Rao Sahib Joseph, T.C. Joseph, was the first medical graduate from Thachenkary. And he was very well introduced by from the comparing desk. And he retired from service as the district health officer of Nellore of that time Madras Presidency. And he was honored with the rare title of Rao Sahib in 1928 by Lord Irwin, the then British Viceroy and Governor General of India. And after his retirement, he returned to Thachenkary and lived here, and he died on 8th September, that is this month, 8th September in 1948. Let us all thankfully remember his contributions to our nation, and his nephew, his generous contribution for this speaker, Dr. Cheriyan's generous contribution to this speaker series. So this is a special occasion for me to welcome, and I am very happy and proud to have Dr. Thachenkary Singh in our campus, and a warm and hearty welcome to dear Professor Thachenkary Singh to this speaker series. So here we have with us Dr. Jayendran Sir. He is a very good friend of St. Berkman's College in many ways. Though he is from the university, from our mother university, that is Mahatma Gandhi University, he is our academic council member. And all the more, he was in our campus many occasions. On many occasions, like for the extension of our autonomy status, and for the many other occasions, and doing some academic deliberations here also, he was here in our campus. So the very good friend of our college, Dr. Jayendran K, he is with us, and hearty welcome, Dr. Jayendran Sir. And on the dais, we have Dr. Shinu Waki, convener of this symposium series. He was working, or the person working behind all these academic activities in connection with this speaker series. From the first day onwards, that means when this speaker series symposium was instituted, I just asked him to take charge of this speaker series. He was very willing and happy to accept that responsibility and was doing a lot of things in connection with this speaker series to make it happen in our campus in the best way possible. So a very hearty welcome, Dr. Shinu Waki, the convener of Symposium series, to this function. Dr. Ranjit Thomas, you all know him as the head of the Department of Chemistry, and you know he is renowned as one among the 2% scientist list published by the Stanford University. Consecutively for the second year, he belongs to that list. So we are proud of him having in our campus and doing all type of research and publications. So he is the secretary of this symposium series. Hearty welcome, Dr. Ranjit, to this function. Dr. Joseph Job, the vice principal of our college in charge of academic matters, is here with us. He is instrumental in making this symposium series, the first one, making it happen. And you know, he is here with us in taking many great initiatives which are happening in our campus in the very best way. He was there in the forefront to run that exhibition, our Summit 2.0. He was there in the forefront, and he was truly, we all know that he is a great leader, and his leadership capacity or qualities make all things happen in the best way possible in our campus. Hearty welcome, Dr. Joseph Job, to this great seminar. So we have with us our teachers who is eagerly waiting for this symposium. As our eminent resource person already suggested, fight against microbial infections using innate immunity is a general subject which, though I am from the Department of Malayalam and not from the stream of science, anybody can easily understand. So, but here we have our teachers mostly from the science department. That is, from microbiology is there, from chemistry is there, from biotechnology is there, then botany is there, and from zoology, physics, from all these science departments, teachers are here. Hearty welcome, dear teachers, to this symposium. And here we have with our dear loving students. Dear loving students, this is a great occasion for you to hear one of the world-renowned biophysicists. And I think you are blessed to hear this type of great persons in the campus. As I said in the beginning, SB was always in the forefront to bring this type of great personalities to our campus. And all who take admission in SB College get that privilege to hear this type of great personalities. So dear students, this is a great day for you, and I wholeheartedly welcome you all to this great symposium. As I said, this is one of the greatest days in our campus. So I wish all of you to have a great day and a great occasion. I remain. Thank you.
Dear distinguished guests, faculty members, and students. It's my great honor and privilege today to introduce a distinguished scientist and scholar whose contributions to the field of biophysics and structural biology have left an indelible mark on the scientific community. Our esteemed guest, Professor Thachenkary Singh, currently serves as the SERB Distinguished Fellow and Distinguished Biotechnology Research Professor at the Department of Biophysics, All India Institute of Medical Sciences, New Delhi. He also holds the prestigious position of the first chair professor in the name of G.N. Ramachandran at Mahatma Gandhi University also. Born in 1944, Dr. Thachenkary Singh's journey in realm of science began with PhD from the esteemed Indian Institute of Science, Bangalore. His career has been marked by significant milestones, including a postdoctoral fellowship under the tutelage of Professor Robert Huber, a Nobel laureate, during 78 to 80. Upon his return to India, he served as a leader at the Sardar Patel University and as an additional professor at the All India Institute of Medical Sciences, New Delhi. Dr. T.P. Singh is renowned for his groundbreaking work in rational structure-based drug design, structural biology of proteins, and X-ray crystallography. His research has had a profound impact on development of drug design approaches in various areas, including antimicrobial therapeutics, tuberculosis, inflammation, and gastropathy. Notably, he is the first Indian scientist to have received all the 6 Ramachandran Awards in the country, a testament to his exceptional contributions to science. His academic and professional achievements are reflected in his fellowships and memberships in prestigious academies and societies, including the Third World Academy of Science, International Science Academy, National Academy of Sciences, Indian Academy of Sciences, Alexander von Humboldt Foundation, and Biotech Research Society of India. Professor Singh's dedication in the advancement of science is further demonstrated through his editorial board memberships, where he has served in key roles. He is a member of the International Journal of Biochemistry and Molecular Biology and has held the position of chief editor of the Proceedings of International Academy of Science, New Delhi, and additionally, he has been member of editorial boards of Indian Journal of Biochemistry and International Journal of Physiology, Molecular Biology of Plants, and also in the Indian Journal of Physics as editorial board member. His contributions in these journals have enriched the scientific discourse. In addition to numerous fellowships, memberships, and editorial board roles, Professor Singh has received numerous awards and honors, such as honorary Doctor of Science degree from the Karnataka Open University, distinguished professorships of University of Mysore, distinguished lecture award of Andhra Pradesh Science Congress, Professor D.M. Bose Memorial Award for Bose Science Institute, Calcutta, and the Thachenkary Memorial Science Award for Science and Technology, among a few others. His work has been recognized both nationally and internationally. Professor Singh's impact on scientific community is further underscored by his impressive publication record. He has authored 339 papers with the H-index of 43 and more than 5,000 citations, according to Scopus Index. Ladies and gentlemen, please join me in welcoming the distinguished Professor Thachenkary Singh to inaugurate the first Rabu Sahib Dr. Joseph Thachenkary Memorial Distinguished Speakers Symposium series, later on what promises to be a remarkable and insightful lecture. Thank you very much.
So it's a great pleasure for me to be here in whatever way. I'm really very delighted. I thank you very much, and I'm so happy. I will make a little bit of gist of our contributions and try to speak in a very simplified manner so that a professor of Malayalam can also understand. But I should tell you one thing, that this part of the country has produced many great scientists, and I will name a few of them whom I have interacted with very closely. And I think first and foremost is Professor G.N. Ramachandran, who was born in this part, in Kerala. All the people talk about Nobel Prizes, but many scientists make contributions which are even higher than Nobel Prize. So he was born here. Then I have another friend who is a very great scientist, currently working in USA, Professor John Kurian, fantastic scientist on structural biology. I also know one Professor E.C.G. Sudarshan, who also missed a Nobel Prize by some kind of politics, who was visiting permanent professor in IISc Bangalore. He was also from here. And of course, my mentor, Professor M. Vijayan, has made enormous contributions. And my another teacher from IISc, Professor K.P. Gopinathan, and many others, Jayachandran was telling. So this part of the country is highly, highly educated region of the country, and has produced many, many great scientists and many writers and poets. And I was also lucky, in 2001, I received one prize where there was another colleague from Kerala, the filmmaker, I think Mr. Gopal Krishnan, through somebody, a film producer from Malayali films. He was also there. And I was very happy to be with him. So I'm very delighted, and I love this place and people from here. So thank you very much. And I will talk in some simple terms so that a professor of Malayalam can appreciate at the end of it. I will find out that. Thank you so much. So you see from the topic, you have heard about vaccines, antibodies, antibiotics, and other antibacterials or antiviral drugs. But we don't talk much about innate immunity. This is the first line of defense your body presents to fight organisms which enter your body. And we thought that innate immunity is a kind of thing, a germline stuff, it is there, you can do nothing about this. But our investigations, we have found out that innate immunity can also be exploited for therapeutic applications against infections. So we'll see how we go about this. So this thing, he taught, by the way, he examined the thesis of Ada Yunath also. Her thesis came to him for examination because she was also, her supervisor was working on collagen in Israel. I think you can read it for yourself. India's greatest scientist as far as contributions are concerned. And you see his greatest contributions correspond to this, the structure of collagen, a 5-side map, and beta turns. These are fundamental. These are so fundamental today. We cannot do any scientific discovery in protein science as far as structures are concerned. You cannot do drug discovery today without the values provided by him. So it is so enormous impact on our current situation. So in 17 years, this is particularly to address Father Kurian's personality and to tell him that how important it is to be a principal, vice chancellor, or director of an institution. He spent 17 years in the University of Madras. There was nothing in those years, 1950s and 60s, early 60s. And he made this contribution, this enormous contribution, with very primarily his own capability. He expressed in such a way, using very simple tools, and he determined the structure of collagen. That was the first time anybody talked about the structure of collagen. He discovered that. He gave Ramachandran's 5-side map, which is fundamental today for all of us to do protein structures. And his group also gave beta turns. These are specific when protein chain folds, certain turns happen. They were defined by his group. So these were enormous contributions. But what was fundamental to this? The location, the geography of the city, the Vice Chancellor who gave him unlimited freedom, and not just freedom, at least you create an ecosystem. The way you were talking good about your colleagues and students, I think Vice Chancellors and principals play a very important role. They are not principals or Vice Chancellors. They should be called as mentors. So you are a mentor in that context. If you can encourage your colleagues, encourage your students, they will deliver very nicely and very well. So Vice Chancellor there at that time encouraged him so much that he could make so much contribution. There were only 3 centers in the world. One was in USA, one in UK, and one in India. That was in Madras. So you can imagine the magnitude of his quality research. So this is just, I always say this to him because what we have done in our career, we have used his contributions as the basis. Now coming back to this, we all know that we all have experienced, I think you are still experiencing in some part of Kerala, some virus in Calicut and something happened. Jayachandran told me something happened in the hostel of MGU. The viral infections, bacterial infections, they are very rampant. Sometimes they are of the nature of pandemic that they become enormous and they cause so much damage to mankind. So here you see what is the, okay, you can read from this that which are the entry points for these organisms. They enter through your nose, your mouth, eyes, and wherever there is an open part in your body, they enter, and that's how they cause damage. Now, whenever these organisms enter your body, and through whichever route they enter, at the entrance of your body, certain proteins are expressed. And these proteins primarily are expressed to fight these organisms. Now, their expressions rise when the infections increase. So, these are the proteins which provide the first line of defense. These are the proteins of innate immune system. In principle, they should be able to protect us, but we are not protected, so we will see why we are not protected, and that would be the thing we will see here. Now you see, this is a very, all of you have seen that when this coronavirus is spread all over the world. And many, many countries which thought that they were very strong in medical sciences and modern biological research, I think they suffered the most. The countries in Europe and Western Europe and USA were devastated. And this was a pandemic all over the world. We couldn't handle the situation. Now, why couldn't we handle the situation? These are the questions we need to ask, and how can we handle this so that these things don't happen in future? Now you see, this is the way that you can read it here, that what are the ways we can fight these infections. So as I said, the first and foremost are innate immune proteins, the proteins which recognize these invading microbes. When these foreign pathogens enter your body, these proteins identify them by the nature they have, the surface, the structure they have. And then they interact with them, they neutralize them. They should theoretically be able to neutralize and protect us. And of course, they are doing all the time. See, many times you are infected by these organisms. You don't come to know, because these proteins have taken care of that. You only come to know when they fail, or when the virus or bacteria overcomes their actions. So all the time we are infected by these organisms, but we don't know whether we are infected or not, because these proteins of the innate immune system are protecting us all the time. So we only notice when they are not able to protect. So you should understand this, that how important these proteins are. So what we want to address today is, why are they not able to protect when we are infected? In principle, they should protect, and they protect all the time. But in certain situations, they are not. Why? That is the question. Now, when you are infected, the next step is that after a while, after some days, specific antibodies are expressed to fight those that particular organism which is infecting you. And to support this, we can also make vaccines so that all of you are prepared, the antibodies are already generated, and you are protected. So there's a second thing, this adaptive immunity. And the third, we all know that we all take all the time antibiotics. These were discovered long, long ago, maybe 70 years before, or that kind of period, maybe 60 years before. Now, these antibiotics were very potent compounds, and they protected us against a variety of bacterial and viral infections. And we were very happy that we had some drugs which could protect us all the time. But now you would see that they also lost their potency because the organisms have changed the proteins where they were binding. And the last, of course, is synthetic antibacterials. That is what the scientists and pharmaceutical companies produce in the labs. But since antibiotics were very powerful compounds, very potent molecules, we didn't make much progress on this. But this is how you should see that. So these are the 4 ways, as of now, we can think of protecting ourselves. Now, I think this is an important message, as Father said, that this institution was created in the 1920s. See, these things, when they happened so many years before, is also an indication of a developed civilization. That when your mind is developed, you are thinking about education, you are thinking about new concepts. So in this context, if you see, this part of the region where the arrows are shown, the civilization occurred before. And that is where, imagine when we did not have antibiotics, we did not have any idea of molecular science, we had no drugs of this kind. How were people surviving? How were they protecting themselves from, because these organisms were there, they were infecting people. A lot of people were dying at that time also, but still, they discovered or they identified certain substances. If you can read it here, you can see all kinds of rotten stuff and many things and by way of observations, they determined that these were beneficial against certain infections. And that was primarily done by civilized minds, the minds which could think. If your mind is properly organized, you can do wonders. So even those years when there was no science of this kind, what we study today, they had found out certain materials. And this was happening in this part of the world. I thought if a computer was here, I could have shown that, but nevertheless. So you just see that where this was happening. But then, you see, when advancement has to occur, you have to not only have your brains, your minds, your civilization, you need technology also. And if you see the next slide, now the location you have changed. Now this is a different location, geography is different. Previous geography was different, the new geography is different. But this part of the world, they had technology, they have industrialization. So they could identify what these compounds, what these materials contain. You see the first one, if you see, he prescribed what was used in the previous locations. And then they did some more observations, like the first antibiotics was discovered. You can see that also. A scientist, today we talk about good laboratory practices, and we want to be very careful. No dust, no shoes, no organisms, very protected lab environment. But earlier times, science was more of a passion, not directed in any which way. So this man, Alexander Fleming, very careless scientist. You know, afterwards, when people are given Nobel Prize by some discoveries, we talk so big about people. But actually, if you see them in the real life, Maybe some students are more serious than them. So he was very careless. He was growing a culture of bacteria. He left the dish open in this kind of environment. And then when he came back, fungus grew on this. We see this all the time, fungus growing on when things start rotting. But if you are a prepared mind, if you understand, and if you can think, then you noticed that wherever fungus grew, bacteria died. So he thought there is something in this fungus which is killing bacteria. But he did not know how to find out what is there in this fungus. So he went to a neighboring chemistry lab. There is a very famous chemist here, Dr. Ranjit, I think. Where is he? Somewhere. So he went to the chemistry lab and requested him, can you isolate what is that which is killing this bacteria? And that's how a compound was isolated and identified, and that was penicillin, and that was the first antibiotics. So scientific research grows like this. Previous location did something, this location did something further, and then other locations will do something further. So scientific research globally progresses like this. It's a global nature. It is not that we are doing some research which is good for India or somebody is doing research which is good for USA. I think scientific research is good for the humanity as a whole, is for the world as a whole. So we have to think about what we do for the world as a whole. And until our scientific research is not competitive globally, it has no meaning. Because then somebody is already doing things better than you, and your work has no meaning. So we have to stay at the front line, and that is what is importance of global programs. So you see that this is inspiring. You should be very proud that you, to begin with, gave the stuff which contained antibiotics. Somebody else there at different location isolated and identified antibiotics. So Indian minds that way are very, very good minds. Now you see that you have to also ask many questions, what is going on? See, we suffered so much in the pandemic. Antibiotics lost their power because they developed, the organisms developed resistance to them. We did not understand innate immunity, and our innate immune system was not able to fight. We don't know why. We should know why. And then adaptive immunity takes a while. Vaccine cannot be developed very fast. And scientific community did not make many antibacterials. They were complacent. When antibiotics came, they were so good, the people thought, pharmaceutical companies thought there is no need to do anything else. They were very satisfied with antibiotic. So not much has been done. You can see very few compounds were made in something, very few compounds came as a synthetic antibacterials investigated and discovered by scientists. So that is the status. I'm just telling you how poor it is. Now, if you see from this slide, it is clear that we were very, very happy with antibiotics. And then in 1990s, around that period, we started talking about resistance to antibiotics. It might have been happening before, but we noticed only in 1990s. You see, when you take a drug against these organisms, and when you take it repeatedly, organism realizes that this is something killing it. So it will modify those sites where these drugs go and bind in them. So they start mutating, they start changing their structures. And that's why resistance develops. So it might have happened before 1990s, but we started talking about this in 1990s. And now we can say that we are in the same pre-antibiotics era, in the sense that we cannot rely on antibiotics. Making vaccines takes time. Developing antibodies, that immune system in your body of that kind takes a while. We don't know anything about innate immune system. We have not made many synthetic antibacterials. So where do we go from here? So again, I came back to this. since you have the second, third, fourth things you have been trying, what do we do with innate immune system? Can we do something? Can we invoke innate system? Can we strengthen the innate immune system? Do we understand this, how it does? And can we find out ways and means to use innate immune system to fight infections? You see, your innate immune system is a response to the quality of your body. If your body is not good, there are many ways your body cannot be good. And if it's not able to protect, we should understand what is going on. So that is the thing we will talk today. Now if you see this is just the thing I said in the beginning, that these organisms, they enter your body through nose, mouth, and all other where there are openings. So in your nose and mouth, there are these mucus and these saliva and all those things. That is where certain proteins are expressed. And they are the ones which fight. For example, you have some proteins, well-known proteins like lactoferrin, lysozyme, peptidoglycan recognition proteins, and then transferrin, lactoferrin here, and lactoperoxidase here. So these are commonly known proteins, but we didn't realize how they fight and how they could be exploited, but they are part of the innate immune system. Is this clear? So then we looked at all these proteins which are highlighted in yellow. We have studied them in details. We have done their structures, and we have studied them from all kinds of animal species. And we have understood now how these proteins of the innate immunity can be exploited to fight infections. So this is the question here. Now in my group we had worked on large number of protein things, but the one I will talk about this innate immune proteins, and out of this only about one protein, and you will see that how you can exploit this and how you can overcome these problems. You see, when you talk about these members of the innate immunity, they use different mechanism of neutralizing bacteria. See, it's something like first line of defense. You have, like your border security force, they use different kinds of weapons. So it's not only one type of action they have different actions. One of them binds to cell wall molecules of these organisms and then neutralizes. Other one removes iron from the environment, deprives bacteria of iron and then virus, and then they die. And the third one hydrolyzes peptidoglycan, the cell wall of bacteria. And the last one is an enzyme. What it does that, it converts certain metabolites in your body which work as substrates into highly antiviral products. So we will talk about the last one and how we can take care of mankind. So this is, of course, on this protein we have. Now, when you do a protein structure, Ramachandran did a structure in 1950s of collagen. He did by modeling, way of modeling, and collagen structure is a helical, triple helix. But most proteins are very complicated in their structure. They have a helical structure also, non-helical structure also, extended different types of elements of structures. They are made into a globular form. So when the first time structure is determined, ab initio, using experimental techniques, this is how it looks like. And when you look at this, even the very educated biological scientists or biochemists or chemists, by looking at this molecule will say, what is this, some sort of silly looking thing, we cannot make out anything out of this. So structures are very complicated. They are globular in nature. The proteins are very large molecules, but they develop a cleft which works as a binding site which produces the function. So rest of the structure is required to maintain that cleft. It's like you make a lock, very big lock, and the keyhole is very small. And the keyhole operates with the help of a key. The hole, you would wonder that why to make such a big size of lock. Bigger the lock, stronger it is. So you require that structure. So from this you cannot understand anything, but to make it simple for audience like this, if you see that we make this in some kind of figure, and you see some cleft now emerging. You can see a cleft on the right. That is where this is an enzyme, a small molecule goes and binds and gets converted into a product which is antiviral, highly strongly antiviral. So like this. Now you can see a little bit more on this. I'm not going to spend much time on this. Then you see I still magnified that, the binding site, how it looks like. Then you can schematically draw this channel. In a large protein, a channel like this is where the compounds enter and interact. So only those compounds which can enter into this and have complementary chemical composition can interact with this protein. So this is how it goes. To see more, I mean, this is just to show you how these clefts are made in proteins very nicely. Now this is a protein, a large protein, but the binding site is very small here. Very small here on the right side, what you see that there is a W1 water molecule, and that's where it binds. So I don't spend more time on this, but I will come back to something else. So this protein, how it does, I think you were saying something about beta glucosidase, somebody works here. So in your body, there are many substances produced as metabolites because of requirement of several biological processes. So one of them very commonly produces hydrogen peroxide, H2O2. Hydrogen peroxide is required by this particular protein of the innate immune system to produce what we call as the intermediate. And then the actual substrate, which is physiological metabolite again, thiocyanate, is converted by this enzyme into hypothiocyanate, which is highly antiviral. So in this reaction, if we have enough of enzyme LPO, if you have enough of substrate H2O2 and enough of substrate thiocyanate, you will have no infection at all. So what is lacking? That is what we have to answer things. And the scheme is like this in a simplified manner, that you have H2O2 produced, then you have SCN enzyme converts this into antiviral products. The issue here is this SCN, H2O2 is in abundance in human body. SCN is not governed by the state of infection. This is a normal metabolite. Its concentration may not be guided by infections, but enzyme is highly, highly expressed. So enzyme is there, H2O2 is there, thiocyanate is not there. So if you can find out a replacement for thiocyanate, then you can cure, you can protect yourself from viral infections. So I think I will go to this very common observation, keeping the audience in mind. Professor Kurian, these are the points, when pandemic was rampaging, everyone was coming with some kind of models that this, you heard television talks, you heard a statement, you heard writings on newspapers and magazines that, okay, what was that? You see, somebody said, not somebody said, it was observed. Epidemiologically, somebody made the data that smokers were protected from this viral infection. So this is a kind of, you would wonder that smoking is bad, but it was protecting you from viral infections. We have to find out why. And then, primarily because it contains thiocyanate. In the tobacco leaves, there is a good content of thiocyanate. If you are smoking, you are swallowing thiocyanate, and substrate is being available. So you are producing antiviral things. And then, of course, we also found out that then people started administering potassium thiocyanate as a supplement. It didn't work. Smokers were protected because of thiocyanate. When potassium thiocyanate was given orally, it did not help. So these were observations. They needed scientific validation, scientific way why this is not happening. And then you see there were observations, many data came, that Japanese guys were not infected by coronavirus as much as people in other parts of the country, other parts of the world were infected. Then people said all kinds of things about Japanese people. But the crux was that they consumed seaweed's diet, the diet which contained high concentration of iodide, the iodine. So that could be the reason, but people made all kinds of theories about people. And then, you see, next, potassium iodide was given, it didn't help. If iodide was contained in seaweed diet, why potassium iodide did not help? So they were matters of scientific. Then patients who were on nitric oxide support. Nitric oxide is given for vasodilation when you have problem with your vessels, your blood vessels. Nitric oxide is given to you. Now it was observed that those who were taking nitric oxide support, they were not infected by coronavirus. So again, there is an important observation. And then the sixth one, patients on TB treatment, those who are undergoing tuberculosis treatment, they were not infected by coronavirus. Now, so there is something in the TB that what you give, the combination of drugs, and the compound INH, isoniazid, is one of the compounds. Could that be a critical factor? And then the last one, the women folk were infected less than the men of their age, and that could be a single factor like estrogen. Now, all these molecules and agents present in these states, you have to find out, not make hypothetical statements and anything, why they were critical, how they were used by the system, that the people who had these things were protected. So that is a very common thing, but important point. Now, what we've made a statement that this particular enzyme, this LPO, lactoperoxidase, it requires, it's a substrate, it requires hydrogen peroxide, which is there, it requires a substrate, and converts this into antiviral, antibacterial product. Now, all these compounds we listed there, are they really working as substrates for this protein? And this protein is part of the innate immune system. This protein is highly expressed when you are infected. So that one has to determine. So we did structures of this protein, and in many ways, without anything, the binding, I don't want to bore people like Professor Kurian in this, so I will skip these slides. So then you determine how hydrogen peroxide actually binds to this protein. You have to show what you have observed by common sense, you have to scientifically observe that. And we found that hydrogen peroxide binds to this protein nicely, and that is required, the first step. Then we want to answer this question that if smokers were protected, we assume that because this contains thiocyanate, if this protein has to function. But potassium iodide did not work. Why? And potassium thiocyanate did not work. Why? So these are the first two things you see. And then what we did the structure with this thiocyanate, and we found by structural experimental investigations that it converted thiocyanate into hypothiocyanate, what you see it here. So it is proved that if thiocyanate binds to this protein, it is converted into hypothiocyanate, which is antiviral, and that is how it protects against viral and bacterial infections. Then, why didn't it do when we gave potassium thiocyanate? And the reason was that potassium in this bound to the site where substrate should bind and block the site. So you should think about different metal ion or different form of thiocyanate to be administered, not as a salt of potassium, in the sense that that by doing these experimental investigations, you prove that thiocyanate is helping you to fight infection, where it is converted by this enzyme into hypothiocyanate, which is antiviral, antibacterial. So that first point proves. And the next point, you prove that potassium thiocyanate is not a good choice because potassium inhibits the binding site. So you are understanding what is going on. So you can take care of producing the right kind of molecule which could work as a drug. So these are very common observations which we scientifically establishing experimentally. And that is the importance of this work. Now next is that Japanese people remained unaffected by this virus for a very long time. Of course, later on they were also affected when there was too much there. But initially they were less affected. And a lot of people said a lot of things. But we said it may be due to iodide in their diet because we were working on proteins of the innate immune system, the proteins which can convert compounds into antiviral agents. So we thought that does iodide bind to this protein and gets converted into hypoiodide, which is antiviral. So we're actually conducting experiment scientifically and proving that yes, it does. But when potassium iodide was given, it wasn't, and that is like previous things. So just to prove that, now you can see how the iodide content of the diets of different locations is. The recommended one by WHO, you must take 1100 micrograms every day. But what in practice we are taking is much less. You can see that people in the US take very little, Indians take little more than that. So Indians were protected better. Less people died from India than USA. And if you see Japanese people, they are taking 500 micrograms, more than Indian people, but it's still not as much recommended. So this is a good correlationship. And if you see the next slide, you can see from this data, this is very authentic data from some international site. In the last slide, you see wherever people were taking more iodide, through whatever way, people died less per million population. You can see the last column. So this is these things, what you observe, what common people do epidemiologically or what common people talk, they can be scientifically established and then introduced as medicines into market. That is how drug discovery progresses. So then we did that iodide thing, and iodide binds to this, and the next structure proves that this enzyme converts iodide into IO minus, hypoiodide, and that is what is antiviral, and that is what is protecting people. Now the next one, if you see, potassium iodide, same story. Now, thousands of cartoons appeared on the scene all the time when this. A lot of people were making a lot of publicity and a lot of name for themselves by talking all kinds of things. So some scientists somebody made this cartoon particularly to suit our observations that when you talk about these hypothetical models, you say maybe this, maybe that, maybe you add this, you subtract this, all kinds of modeling things were going on. People of mathematics were making models on coronavirus. All kinds of people were contributing. But actually, there was no clarity. That's what this shows, that if you can read this, the clarity is not there. Clarity only came when we talked about iodide actually being converted by this protein that way. Now next, nitric oxide. Nitric oxide support is given to patients who have these problems of vasodilation, vasoconstriction. So that's a common use. But they were happily not infected by this virus. So nitric oxide could also be a substrate. So we did a structure with nitric oxide of this protein, and we found that nitric oxide is converted, NO is converted into NO2, which is antiviral, antibacterial. That is how this protein is contributing. Now coming to next slide, this organic molecule I will skip, but if you see that patients on tuberculosis treatment, they were not infected by coronavirus. These are the facts, epidemiological data which were made available. So what is happening there? What is happening here? This INH, isoniazid, is binding to this protein very nicely. This is experimental data. And you see how it is binding very nicely. So INH is converted into an antiviral product, and that's how it protected. The next is how women were protected, and women were protected, we thought because they have estrogen. And then we examined whether estrogen binds to this protein and gets converted into a product which is antiviral. So when we did the structure of this, and estrogen binds very nicely to this enzyme, and it is converted into a product. So that is why, so the whole story now is like this, that we have certain observations now. We found out that thiocyanate is a substrate, it's a metabolite, its concentration is not in your control, it is not triggered by infections, it's a normal metabolite all the time happening in your body. So you have no control on the concentration of thiocyanate. Now, could you provide thiocyanate externally? The best form to provide this smoking. But potassium thiocyanate is not a good salt. We can think of some other salt, but we'll have to examine those salts which don't block the binding site. So thiocyanate supplement is one way. So diet is very low. So I think tobacco leaves chewing can be a good idea, but we need to make compounds. Now next, nitric oxide. No, iodide. Iodide, one good way was that you could recommend the iodized salt or other iodized compounds which could be given, and iodide is converted by this protein into hypoiodide, which is antiviral, and the nitric oxide, which is converted into nitrite, and that's also antiviral. So these administration processes can be determined, but these were identified as antiviral agents. And then, of course, isoniazid, that INH which we give for tuberculosis treatment, can also be recommended as a drug against viral infections. And of course, estrogen can also be given. And then we have made some organic molecules, chemistry. So that is, they have to go through other processes of this drug development. That is a longer process. But these are the known things and can be administered. So this is what situation now we have. Now this scheme what we presented in the beginning, only SCN, and SCN concentration is a problem. But now we can say iodide can be given, nitric oxide can be given, isoniazid can be given, estrogen can be given, and these are good substances to protect you from viral infections. And that is established experimentally by actually observing them that these compounds bind to this protein and they are converted into highly antiviral product. So this is very clear that how you can do this. So this is the kind of thing we did during this period. We did many structures, but established this. We have got many compounds, organic compounds also, which bind as substrate. Many of them bind as inhibitors, but substrate binding is different. So many compounds are made that way, which can also be subsequently prepared as drugs. Now the point here important is, Dr. Kurian, the most important point here is that innate immune system is something against which no resistance can be determined, because they have many different mechanisms. Different proteins follow different mechanisms. This protein follows, just requires a substrate. And there is no involvement of viral protein in this, so they can modify nothing. And these all agents have a general effect of neutralizing virus, not by specific binding or anything. So innate immune system is one system where there is no resistance, and these proteins of the innate immune system are overexpressed during infections. I talked about one protein, but other 3 proteins have also interesting stories. So this particular protein just requires a substrate, a proper substrate to be given, and as this protein is overexpressed in response to infections, there is no dearth of this protein. H2O2 is produced in abundance. We need to provide substrates. And these all were examples how this protein can contribute. And that's how we suggested that innate immune system, if you understand, can exploit this to protect yourself against invading microbes. And this is very fundamental in that context. So this is what it is, and then I thank you very much for this is the once upon a time, this is the status of my group, I thank them. So this is what I wanted to convey, the message that whatever we do, the day-to-day observations and the crisis we come across intensifies our efforts to produce how to fight them. And that is what is very fundamental in scientific research. It wasn't possible many, many decades before, but we can do many things in shorter times and protect ourselves. So I thank you very much. I thank you very much, Father Kurian, for inviting me, and Jayachandran for thinking of this, and other colleagues. I'm really, really very delighted to be part of this, and I am seeing a wonderful students' audience. But I would have thought that students can ask questions and clarify doubts and make comments. I think a scientific lecture must always be supported by curiosity-driven points. So from my point of view, if they have any question or comments, or they want to help me how to do better, I think we will welcome them. So very bright students can ask things and can comment, can say good things, can say bad things, can make questions, whatever. Thank you so much.
This event is a tribute to the memory of a distinguished individual whose legacy continues to inspire us all. First and foremost, I would like to extend our sincere appreciation to the family of the late Dr. T.C. Joseph Thachenkary for bestowing upon us this opportunity to remember and celebrate his remarkable life and contributions through the institution of this Symposium series. Thank you all for your goodwill and your presence today here. I feel immensely privileged to extend our thanks to Professor Dr. Thachenkary Singh, a distinguished scholar and an expert in the field of biophysics at the All India Institute of Medical Sciences. We are truly honored to have had an opportunity to listen to his enlightening lecture today on the topic, Fight Against Microbial Infections Using Innate Immunity, Adaptive Immunity or Vaccines, and Antibiotics. Professor Thachenkary Singh's lecture has not only been informative, but also ignited our curiosity and passion for understanding the intricate world of microbiology, immunity, and infection control. He has shared his valuable knowledge, insights, and experiences with us, shedding light on critical aspects of microbial infections and the various tools that are at our disposal to combat them. Moreover, Professor Singh's passion for teaching and research serves as an inspiration for all of us present here today. His unwavering dedication to the pursuit of scientific excellence is a testament to the caliber of educators at AIIMS. On behalf of everyone who are gathered here today, I extend our warmest thanks to Professor Thachenkary Singh. Our sincere thanks to Dr. Jayachandran, Professor, School of Biosciences, MG University, who is also a member of our academic council, who has actually collaborated a great deal with us in the conduct of the lecture today. Thank you very much, Professor Jayachandran. Our thanks are due to all who have contributed in one way or the other in the smooth conduct of the lecture today. Very especially our dear Principal Father Reggie P. Kurian, Dr. Joseph Job, Vice Principal Academics, Dr. Ranjit Thomas, Secretary of this Symposium series. Thank you all. Finally, a big thanks to all the participants of this lecture, teachers and students from different departments of the college, and from other institutions around the college, and our dear alumni of the college. Thank you all. Once again, thank you all for your presence and participation in this meaningful event. Wish you all a very wonderful day today. Thank you.